So the first in Google, go and type UniProt. So UniProt is a database uniquely for protein, and it is a database which contains your protein name, gene name, organism, and whether it was reviewed or unreviewed. The protein, whatever we are taking, it was completely reviewed protein or unreviewed protein, and it has annotation levels, whether the protein was completely annotated or whether it was at experimental level. All those information will be available in this particular database. You can also have different forms of data such as your subcellular location, pathology and biotech information, and your organelles, whatever information that is biological information related to complete protein, whatever target you are choosing. All the information will be available in this particular database. So this is the home page of this particular database. So I have given a link in the manual. So if you are working simultaneously, you can click on to that and you can open the screen and you can type any protein of your interest. Usually how we choose protein is depending upon your disease mechanism. For example, suppose if I am choosing cancer, so what I will do is I will search for a mechanism of that particular cancer. And I try to find out which protein were overexpressed or mutated in that particular cancer. So based on these conditions, we choose the drug target, that is your protein. So after finding that protein keyword, so this could be usually done by the literature review and by studying the pathway mechanism pertaining to the disease. So after which we choose one particular drug target. Finally, we try to check whether the particular protein have the structure so that we can proceed with the docking analysis. So first step is once you found the protein, you try to put the protein name in your search box, whatever given over here. Hope my screen was visible for everyone. So I'm searching for GPCR protein. So sir already told about GPCR protein, G protein coupled receptor. So I'm going with a particular protein. So you have to type in the search box and click on to the option search. So where it will list out the protein, that is this particular protein might present in different organisms. So this GPCR and the organism were listed over here. Hope my screen was visible. And if I'm too fast, you can put it in your chat box, and if any doubts or queries in between, you can also put that also in the chat box so that I can clarify then and there. Okay. And so this G protein coupled receptor, so once you find the details, so suppose if I want this particular protein from Homo sapiens, I need to click on to this particular entry. So why I am choosing UniProt instead of NCBI is UniProt is a curated database where they verify all the, so because in NCBI you may found multiple entries for same organism, whereas in UniProt there would be one particular entry for this one particular gene name. So I'm clicking on to this entry link. Hope everyone see this entry icon where you have a link Q5T6XY. So I'm clicking on to that on the particular link. So here this is the output page for this G coupled protein receptor from Homo sapiens. So where I could find the gene protein name and the gene name related to that, and Homo sapiens, that is the organism. Then whether this protein annotation level, whether this protein was reviewed, annotated, and experimental evidence at this particular protein. So whether this protein was experimentally proven protein. So all the information were available in the starting of the page. And when you go for publishing, suppose if you are going to work with this particular protein and you want to publish this data, so you have to use this unique ID, this UniProt ID, this Q5T6XY. This is the unique ID for this particular protein. So when you go for publication purpose, you require this particular ID. So you have to mention GPCR from this particular ID from UniProt database. You have chosen this particular target. And next is your functional information. That is all the functional related, that is this protein plays vital biochemical mechanism, molecular function, biological process, many function it plays. So all the informations were available in the function icon, and apart from that you have other names and taxonomy where this protein might be used in different names. So in some article they may use as HGPCR33, in some article they may use as GPCR33. So based upon the different names. So you can find various names of the particular protein over here. And again, this is your taxonomic lineage of Homo sapiens. For any organism, it would be available. And this particular gene, it was present in the chromosome number 6. So chromosome number 6, this particular gene was expressed. And next one is organism specific database. That is a different, this particular gene, whether it was present in different organisms. So organism specific database information were available. Next comes your subcellular location, where this particular protein will expressed. That information was available in the subcellular location. So here it was given as cell membrane and membrane protein. GPCR is, it is a membrane protein. From the name itself, it was a coupled receptor protein. So it was present in your cell membrane. Next is your pathology and biotech. So pathology, if it is involved in some cancer mechanism, in some protein, the complete information would be present. Some we need to find from literature. So if it was already available and it was reviewed, then they will put over here. For some protein, for example, GLUT4 and all, you can find one paragraph about it involved in diabetes, although information would be available. So based upon the protein you are choosing, this information would be available. And pathology, and here you can see the mutational details, that is mutagenesis detail. Like in this particular protein, that 149th position, the serine amino acid, if it was converted to alanine amino acid, there was a loss of function. So this was reported in some literature. Similarly, another protein, threonine, if it converted into alanine, then the protein would lose the function. So these kind of information will also available for many residues. Depending upon the protein, these information completely vary. And all the kind of sequence related information, that is mutation and pathogenesis and post-translational modification information available in this particular database. Next information is your PTM. PTM is nothing but every protein undergo post-translational modification after the protein synthesis. So in which residues, which modification will happen, that information was completely available here in the amino acid modifications. Here it was mentioned 121. So 121st amino acid, where the glycosylation process will happen during the PTM process, where the asparagine amino acids are involved, and disulfide bond will form in 131. So based upon all the residue, these informations were, so all the complete information were available related to PTM. And next information is your expression system, in which tissue this particular protein will get expressed. That information will be present in the expression database. Next comes your interaction. Interaction is nothing but sir told about network pharmacology, all the initial in the first slide, Giri sir told about the network pharmacological approach and protein-protein interaction. So each protein, it was interrelated and all the data were present in the string database. So any protein, the gene-gene interaction and protein-protein interaction study would present in the protein-protein interaction database. Next comes your protein structure. So protein structure, so for any protein we require structure to analyze. So when you go to the structural level only, you can find the complete you can go for active site prediction, then only you can go for interaction analysis. So you require protein structure for that. So here you can see that this protein have some structure, where you can see this helical structure was there, and in between some beta sheets were present, and remaining things were loops and coils. So all the regions were present, after which you can see here it was actually the length of the protein was 1 to 926. So here in some cases there would be direct link to the PDB database from here, where you can directly download the protein structure from here itself. So the next most important information is your protein domain. So protein domains are nothing but it was a conserved part in your protein, where you can find the protein is this particular part is functional region in the protein. Suppose this particular protein have around 926 amino acids. So in that specific regions are called as domains. So those domain regions you can check using this domain database, when you click on to this information, that is G-coupled protein receptor 3 family, you can check information related to the domain of this particular protein. And next information is your, so these were all the cross-link to the various other domain database. So when you go in detail, so suppose if I'm going to work with this particular protein, I need to know about complete domain details and whatever signature patterns present in this particular protein. So in that case, I'll go and search here. Here you can see PFAM entries. So PFAM, I'll just teach that separately, how we can identify domain using the PFAM database. So next information is your sequence level information. So for any protein, we require sequence. So if the protein has structure, we can directly download the structure. But if you want to know the domain detail, any other features of the particular protein, so in that case, we require the protein sequence. So for protein sequence, you need to click on to the option FASTA, and you can copy the protein. So here you can have a FASTA option, everyone could see, I think so. So here you can have a FASTA option where you can click on to the FASTA and copy this sequence for further analysis. So for any protein, you require a sequence where you can, if the protein doesn't have the structure, you can go for structure prediction. If the protein doesn't have the domain details, you can go for domain analysis, secondary structure prediction analysis, and all the information related to the protein structure. The sequence level annotation can be done using the sequence. So you can copy the sequence and keep it in a notepad for the further purpose. And next is your other information related to the particular sequence. So you EMBL, GenBank and DDBJ, it is nothing but this particular sequence, it was encoded from nucleotide. So there was a link to a genomic database from this particular database. Next is your similar protein related to these protein, and last one is cross references, and apart from that you have miscellaneous information, pathway related information, and so these were all the additional information related to this particular protein. In the last you have the entry information. So initially this protein was deposited in the year December 21, 2004, and it was integrated into SwissProt or UniProt in the year 2005, and lastly it was updated in the September 29, 2021. So this database, it was frequently updated. Suppose this information, this protein related information was published in any article, this information would be incorporated into this particular database. So this database would be kept on updated, and so this is how we retrieve the sequence from the database, and you can get all the functional related information about any protein from this particular database. So it could be an antigenic protein from a microorganism, it could be any protein involved in the disease mechanism or the cancer cell pathways or any protein. So whatever protein you are searching, you just use the keyword and which organism you are going to target. So that alone is enough to browse this particular database. So am I clear? Any doubts in this particular database? Participants, any doubts? Okay. So sequence you can, yes ma'am? Okay. So sequence we can take from this database. So sequence information alone I will take from here. So the next database, next tool we are going to work here is PFAM. So PFAM, as I told you, the functional part, that is protein domain, is important to identify the protein functional part. So we have to go for, in Google, go and type PFAM database. So here you have a pfamxfam.org. So in your manual, I have given clearly the PFAM link was given in the manual. If possible, open the manual and click on to that particular link. So PFAM database, it is usually consists of family, domains, repeats, motifs, coiled, disorder, regions, everything would be predicted using this particular database. the family details. That is, family details is nothing but collection of your related proteins were deposited in one particular family. Next is your domain. Domains are nothing but your structural unit of your protein, where it plays a role in your functional part of your whatever it is. Active site might be present in your domain. So many information related to the protein function would be present in your domain. And repeats are nothing but addition to the domain, you have something called as repeat, where a short unit which is unstable in isolation. So you know very well there would be some sequence repeat. So in nucleotide you may found repetitive sequence. Same thing in protein, there would be some amino acid would be a repetitive amino acid. So that form is called, that region is called as repeats. And apart from that, you have a motif. Motifs are nothing but short unit from outside the lobular domain. And next one is your coiled region, where you can find the motifs and regions that typically contain alpha helix and coils together. And apart from that, you have disordered region, where regions that are more conserved, that is called as your disordered region. I think database was taking too much time here. So let it load. I'll go. So I don't need to waste the time. So I'm going for Pdb database. So in Google, go and type Pdb database. So Pdb database, so you can click on to the option Pdb database, where you can go and search your Pdb IDs of any protein, that is any structure of your study. You can go and search here. If the structure was not available here, you need to take the sequence and you have to go to homology modeling. That we will do it in the next session. So first, if the protein structure was available, how we need to download and how we need to prepare that particular protein for docking. That part alone I'll tell you first. So the protein structures, so Pdb database, it was a unique database for protein data in protein structure, where you have many information related to the structural detail, that is your alpha helix, beta sheet, everything present in the protein structure. And usually this database, in this database you will be having, so sir already told about Pdb database, where it contains X-ray crystallographic structures, NMR models, and cryo-electron microscopic structures were available in this. Apart from that, you have model option also available, where computationally predicted structure was present here. So first, let me put any keyword for the protein search. So I am using BCL2 here. So BCL2 is drug target for breast cancer and some of the cancer. It was a commonly known target for cancer. So I am choosing BCL2 here. So when you put the protein name itself, when you put the gene name search over here, you can have a different page like this, where you can refine your organism. So based upon your organism, you can refine your data, or directly you can choose any one particular structure and you can search for the data. So I'm clicking on to the option Homo sapiens, where it will only show the, and if I click here, I have to click, and here you can see some green color arrow. You have to click that particular icon to get the protein structure. So I'm just clicking on to the option here, BCL2 inhibitor complex. So I'm clicking on to this option so that I can check the structure for this particular protein. So this is your output for your protein structure, where you can see, here you can see the protein name, and you have your protein classification, and next is your organism name, in which particular organism it was present, and which particular expression system was used to express this particular organism. If there was any mutation information, you can see here mutations were available or not. And next one is your experimental data, that is your X-ray diffraction data, where you have a resolution information. When you come from a sequence level, when you found multiple proteins, so in that case you can choose one particular protein with less resolution. So at the next you have literature information, in which particular article this particular protein structural details were available, and next comes your protein name, and here it was mentioned A and C chain. So A and C means homodimers. A chain and C chain is the same apoptosis regulator protein, and B and D chains are nothing but BCL2 inhibitor. So here you have two different structure. One is apoptosis regulator BCL2, which was present in A and C chain, and next one is B and D chain, which contains BCL2 inhibitors. So this we will refine. So once you download the file, if we require apoptosis regulator region of this particular protein, we take only A chain. If we require inhibitor region of this particular protein, so based upon the requirement, we choose the protein. We take that part alone. So I'll tell you download option later. Before I'll complete all the information from here. Next is your 3D view, where you can see the three-dimensional structure of the particular protein entry. Where you can see the protein structure, you can rotate and check the protein. The dot over here, it is nothing but the water molecule. So each protein, at the end of the protein file, you have a water molecule which was attached to the proteins, because it was X-ray diffraction structure. So definitely it has water molecule. So here you have BCL2 protein. Apart from that, you have this inhibitor region also present. So you can rotate and check the protein. In some protein, you might have a small molecule was also present along with this protein details. So here I can zoom it out using the mouse. You can rotate and zoom in and zoom out. Next comes your annotation part of the protein, where you can have a complete annotation related to the structural details, and you can have the domain details, and you have something called as protein classification family, CAT, that is classification, architecture and topology, homology, where you can find the complete structural domain features of your particular protein, and it was classified according to the classification level of your protein. So these were all the functional details pertaining to this particular protein. So next comes your experimental details, where it was completely your X-ray diffraction. So when they done the X-ray diffraction studies, they have used different method to crystallize the protein. So in which method they have done the X-ray diffraction studies, and what is the crystal data size and diffraction pattern, radiation source, and what were the different types of data and refinement data, everything was present in this particular page. So this was completely how the experiment were carried out, under which condition the X-ray diffraction protocol X-ray diffraction methodology was followed. And next comes your sequence information, where you have the sequence data of this particular protein, where which region is domain. So as I told you, like domain would be predicted. So here you can find the domain regions present in this particular protein, and so here you have a PFAM input. So here you can see one pink color region, this one region. So these are the two domains present in this particular protein. So if you click on to this detail, if I click on to this pink color, you can see here it was nothing but BCL2 domain. It was predicted using the PFAM database. So it was the position of this particular domain is from 97 to 195. And if I click on to this particular domain, you can see the position was 8 to 32. Okay, so this is about the protein sequence related information. That is the length of the protein, if it's around 1 to 192, what region was domain. So all the regions, and here you can see different hydropathicity. So hydropathicity of your protein. So based upon this, the increase or decreased hydropathicity would be there. So all the details related to the sequence based upon the amino acid was present in the particular position. Then comes your genome information. So if I click on to this genome icon, I can find the genome information related to this particular protein, in which chromosome, which particular location, which region, this particular. So this arrow depicts in this particular region, this particular BCL2 was available. So it was present in 21st chromosome, Q region. So you know very well, in chromosome you have P arm and Q arm. In Q arm, in 21.33rd position, this particular gene was available. Next comes your different versions of this particular data. Okay, so in which date it was released, so that detail was available. So finally, we can go to the download files, where you can download the PDB format of the particular protein. So this is how we can download the protein structure. So here you can see the protein structure was download. I have already told to install Rasmal tool. If you have Rasmal tool, so you can see here the icon. It was showing. It was actually showing the Rasmal tool. If I double click, it will directly open in Rasmal. Able to see this? And so in Rasmal, you have different forms of things. This is your wireframe model, and this is your backbone model of your protein, and this is your space fill model, where you can, so you know very well it was three-dimensional space. Even if you rotate, it will have the three-dimensional space, and you have ball and stick model and cartoon model, and so usually for publication purpose, in many in many articles, you would have seen this particular form of the structure. And based upon the color option, you can use secondary structure means. It means based upon your helical beta sheet, the color would vary. So here, this particular protein, it contains helical structure, so it was shown in pink color. One more thing I have to tell. So once you want to open the, so once you download the PDB structure file, open it with first with Notepad. So how you will open means, so you have to right click, open with, use the more app, or usually if you have WordPad option, you can use WordPad option to open the this particular file. So here you have this protein name. Apart from that, you have these are all the general detail. It will be present in the PDB database. So it has some format. The PDB database, each line it contains 60 characters. So usually, along with the space, complete length of this particular alphabetical character is 6. So you can have the, so here whatever details initially we are having, it is completely not related to the structure. So when you go in between the file, you can, from the atom position starts, there comes your structure. So here you can see the structural details. So if I am minimizing the file size, you can see it little clearly. I can, now you can able to see, I think so, everyone. So these are all the atomic coordinate files of the protein structure, where you have A chain, B chain, and C chain and D chain. So I have told you A and C chains are a BCL2 protein, and in between B and D chain are the BCL2 inhibitor. So here the atom number starts here. So you can see the threonine amino acid. So from here to here, it is a complete one amino acid structure followed by glycine. So whatever amino acid order we are seeing in the sequence level, the same would be followed in the structural level. And so this is your atom. And what is the atom? That is, you know very well, in amino acid you have different types of atom: nitrogen, carbon, oxygen, and in case of sulfur containing amino acid, you have sulfur group and oxygen group. So these were all the orders of the whatever it is, the amino. So usual amino acid pattern order we follow. And you have x-axis, y-axis, z-axis value. So because you are rotating the data, so you should have all the values for the three-dimensional structure. So you have A chain over here. So once the A chain was over, you can see a B chain. Okay, so hope everyone see this file. Is it visible? Okay.
Yes, ma'am, it is visible.
So you can scroll. Okay, thank you. So finally, if I need only the A chain, I can delete remaining part of the structure. So how I'll do is, so in the A chain, I'll have the termination TER atom. So at the end of each chain, you have TER atom, which is called terminator atom or end atom. So after which I can delete complete structural file over here. So you can do it by scrolling the mouse, or you can use Control Shift N to delete the entire part. And I have already told you, at the end of your structure, whatever structure you are taking, you will be having the heteroatom, that is the water molecule. Here you can see HET atom, water molecule. So that part also we need to remove before proceeding with the docking. So only one chain we will take to the docking. We won't take the entire A chain, B chain and C chain, D chain. Hope I am clear. So I am deleting remaining part of the structure, and I am going to save this structure. So now I am going to open this particular part of the structure with Rasmal. So I am just going to double click it. So now you can see the clear structure of the one particular chain. Okay. So initially we saw 4 chains. So now it has only 1 chain. So this is how we will download the protein structure and we prepare the protein structure for docking. So am I clear? Yes, definitely I'll move it slower. So I'll repeat once again how to download the structure of the protein from Pdb database. So usually in Pdb we will go and search for a protein. This time I'll choose different protein. HER2 I'm using here. HER, it is human epidermal receptor. It is a target for cancer. So here I am choosing the option Homo sapiens, and I am going to click on to the arrow to refine the structure. Um, so here you can see the crystal structure of extracellular domain HER2 complex. So I'm going to click on to this particular structure, and once you open the structure, you need to download the file, download the PDB format of the structure. So once you download the structure, so usually it has different number of chains. You need to check the chain details, all those things before you download the structures and which chain you are going to choose. So this is nothing but HER2 protein. So it contains antibodies along with the protein. So these antibodies are from Homo sapiens. So somebody in morning session they have asked how to download a monoclonal antibody. So all these things are nothing but protein which was available, the structures would be available in the database if the structure was predicted using any crystallographic method. So here you can see this A chain is your antibody, so you can't take that particular chain, and B chain is also antibody, and C chain is your actual HER2 protein. And here you can see the mutation detail and other co-crystallized structure present with this particular protein. So in some cases, we use this co-crystallized structure as inhibitor. So co-crystallized structures are nothing but already this protein were crystallized. So this particular co-crystallized structures would be interacted with your protein. So usually if you download this protein will be downloaded along with this co-crystallized structure where it has the interactions also along with that. So if you usually we remove the co-crystallized structure and we perform docking with our inhibitor. So this is your native inhibitor. Co-crystallized structure, something is it is related to the native inhibitor. Okay, and this is how we download the structure. Once you download the structure, you need to open that structure with notepad. No, sorry, you have to open the structure with WordPad. If I directly click it will open, so you have to click open with and click on to the option WordPad. So once you do, so I have told here it is C chain which is required for us. So we need to delete the A chain and we have to keep only the C chain. You have to delete the A and B chain. Okay, so I'm going to delete the A chain. So usual mouse scrolling and deleting. So you have to move the mouse little bit. You have to click the mouse till the selection A chain and B chain gets selected. So I'll delete till here. So C chain started from here. So till T chain I have deleted. So when you delete, you have to be very careful. You are deleting only the particular chain, not the other chain. So once the C chain were over, at the end of the C chain, you have terminal atom, after which your co-crystallized ligand structural information would be present. So here heteroatom starts over here. So you can see these are all the other atomic molecule which we saw on the home page of the PDB database. So I'm going to delete that also, and I'm going to save the structure. So this is the structure of HER2. We can usually you can visualize with Rasmal cartoon model. You can see the based upon the structure. If I choose, you can see you can see the alpha helical structure, pink color. Beta sheets were shown in yellow color, and white colors are the roots and coils. So clear? Any doubts? Any doubts? You can post it in your chat box or so this is how, if the protein structure was available, we will download usually. If the protein structure was not available, in that case what we will do. So now we move on to that particular part. So before going into practical part, let me give brief introduction about homology modeling. Then I will move to the practical part. Is my screen visible? So if the structure was available, we can download it from Pdb database. If the structure was not available, we use any one of the protein structure prediction methodology to predict the structure of the protein. So it means the protein only have the sequence, it doesn't have the structure. Okay, and so So before I want to tell this point alone before I'm going to protein structure prediction. So when you choose the PDB protein for docking, so you have to check for resolution must be minimum possible to ensure better quality. So the resolution should be lesser if whatever protein you are taking. Next important thing is domain completeness. It should be a complete domain. It should not, so domain if there should not be any missing regions in the domain, and definitely the region should be mutated, or the mutation related information would be available. If you're going to work with disease protein, you have to mutate that particular residue and you can take that particular structure for your analysis. And next one is secondary structure. The side chains were also important. There could be some missing residues in the PDB database because it was a crystallized structure. There would be some missing residues. So the complete side chain and structure should be present. And definitely every protein structure, it has ligand, crystalline, water and cofactor information would be along with the present along with the structural detail. That should be removed before we proceed with the docking. And so this I already told you, the PDB file contains protein, ligand and water molecule. We need to remove the water molecule and heteroatoms and remaining chains, whatever it was not necessary to the protein structure. So what is the protein structure prediction method? So if the protein structure was not available, we need to go for a we need to take a sequence, and what we will do is we usually search that protein. So what it will do, it will go and search for the sequence, it will go and search for the sequence structural alignment. So if the similar structure was available in the database, then there would be a homologous in the PDB. So that is, the structure was somehow similar for that particular amino acid sequence. It found some of the similar structure in the PDB database. That could be done using the BLAST. And next is here, if it was yes, we can go for the homology modeling. So for homology modeling, the structural identity percentage should be the structural identity percentage should be more than 40, minimum 40%. If it was 70-80%, then the accuracy of model would be very good. So once you done with structural alignment, we evaluate the structural model whether the predicted model was good or not. That process we are going to do it practically. Definitely, I'll show you that. And if you can't find any template, in that case there was another method called as fold recognition method, where you, based upon the secondary structural, that is based upon the protein fold, alpha helical structure, beta sheet structure and fold present in the protein, we try to find that similar fold of any other protein and we try to predict the structure. So for these methods, some of the online tool Rosetta were available, where you can predict the secondary structure, I mean based upon the secondary structure and fold of the protein. That particular tool, method, it was used to predict the protein structure. Last method is your ab initio structure prediction method, where if you don't have any structure related features, that is common structure related features, in that case you will go for ab initio structure prediction method, where the complete it was completely depends upon the chemistry part of your amino acid, where you have, it was a completely knowledge based method, where it involves molecular dynamics and molecular all the molecular level and amino acid or the amino acids complete physical based information were available. Definitely, I'll show the, but definitely I'll show all the information practically also, how to do also, I'll just show you. So homology modeling, as the term implies, it was called as comparative modeling. It is nothing but you are going to, from the sequence, you are going to convert that sequence into a structure. So you have amino acid single letter code, you are going to convert this particular amino acid single letter code into a structure, that is using the target protein. Target protein is your input protein, and template are nothing but the template, that is PDB ID, which was choosing. So because some protein, for example GLUT4, it was present in Homo sapiens, it doesn't have the structure, but GLUT1 has the similar fold to the GLUT4. So based upon the GLUT1 structure, we can predict the GLUT4 structure. So GLUT1, it was act as a template for this particular GLUT4. So the target is your GLUT4, that is glucose transporter 4, and template is your glutamate transporter 1. This is how we try to predict the structure using the homology modeling. So this is the overall steps in homology modeling, where you have your target sequence, where it performed so ab initio, so one participants asked about ab initio method. So ab initio method is what was completely knowledge based method, where you have to know each and amino acid complete geometry details, everything you have to know. So it was completely so based on that you can predict the try to predict the structure using some of the online available tools. So there was some online available tools for ab initio structure method, and you have a amino acid structure construction tool also, where you can incorporate one amino acid and you can construct the structure of the structure of the particular protein. So you, once you identify the template, it will perform the alignment. So after the alignment, that is your template target alignment would take place, after which your model would be built, after which you will define your model. So based upon this Ramachandran plot, we will define, we will decide whether your predicted model was good or not. So this red color region is highly favored region, where most of the residues would fall. That is, it was your alpha helical and beta sheet region, where all the residues will fall in this particular, all the amino acid which was present in alpha helical and beta region would fall here. And yellow color is additionally favored region, pale pink color is your favored region, I mean, generously allowed region and allowed region, and this white color regions were the amino disallowed region. So where no amino acid should fall here in this place. Once you model the structure, in that case your no amino acid should fall under this white region. So if the more residues was present in the white region, it means your structure was not modeled perfectly. So today we are going to see about Swiss model tool. Swiss model, it is a web-based integrated service for the protein homology modeling. You can use the building, you can build the protein models using different levels. Building the homology model, so it usually comprises 4 different steps. So you identify the structural template, alignment of target sequence and template structure, model building, and you can evaluate the quality of your structure. So usually we go for automated mode where you will give your sequence and what it will do is it was automated mode. The pipeline using this automatically it will connect to the BLAST and it will search for the structural template, after which you will find it will list out the template. So it will show this particular template have 60%, 70%, 80%. So based upon the ranking you will choose the template, after which you build a model. So you can choose 3 templates also, or you can choose any one template where you can go for modeling. So this method uses a continuous evaluation within continuous automated model evaluation platform. It was called as Cameo. And usually using the template search option, you can choose the template and you can build the model. So this is the home page of Swiss model where you will paste your target sequence over here and click on to the search template option. So before going into search template option, you need to give your protein name and email ID, because in some cases it will take time. So even if you close the window, so once you submit and if you click the model, once you click the build model and if you close the window, your result would be coming to your mail. So for that you can give your email ID. So once you click on to the template search option, it will list out the template over here. Here you can see a query coverage option and GMQE, QSE. So GMQE is nothing but it was a score which was actually it should be higher. If it is 0.85, it should be higher, it should be below zero point 0.1, but sorry, it should be nearer to 1. If it was more, it was actually good. If it was less, the accuracy would be less. And the identity should be more. I told you some protein which was actually, if the structure was available, it may show 100% identity, but you can see here the query coverage would differ. So the identity here, the highlighted region, if suppose I have a sequence length of 200 and 185 only the query coverage. So that is why here it was white and here it was blue. So this query coverage, it shows 100% identity over here. So once after this option, once you choose this one, you have to click on to the option build model, and after which once the model was built, you can click here to click on to this PDB format to download the file. And usually this score, that is GMQE score, should be nearer to 0.81 and this score is 0.84. So this GMQE and QMen, this score should be between 0 and 1. If it was higher, nearer to 1, it was expected to be a good quality, and Z score should be less than minus 4.0. So that I'll show you practically when we do. After which here you have something called model assessment. Before this icon there would be something called model assessment. So if you click on to this, you will have this Ramachandran plot where you can see most of the residues will fall under this particular region, green color region, and here there was no regions in the white region. So there should be Ramachandran outlier should be 0% and most of the residues should fall in this favored region. So if the more outliers were present here, then the structure predicted by you is not a good quality model. And if this percentage also, this dark green color is your most favored region, this pale green color is your additionally favored region, and this is your light blue color is your generously favored region, and this white color is your disallowed region. So if more residues fall outside the most favored region also, there would be some quality level. So this minimum it should be about 85 to 90%, this most favored region should be. So then you can use this result for your publication purpose also. If you are going to work with any using this structure prediction method, you need to give your Ramachandran plot result also. So next comes your Rasmal visualization. This I have already shown you. This Rasmal is your molecular graphic visualization program that was developed by Robert Sale, and it was an open source platform to visualize your protein structure. So let me start the practical session for this particular homology modeling, where I require a protein sequence without the structure. So I'm going to use GLUT4 protein for this purpose. So I'm going to take the sequence of GLUT4 and perform the Swiss model server. So I need a sequence from Homo sapiens. So I'm clicking onto the first option, and I require FASTA format. So I'm clicking here and taking the FASTA format of the sequence from here itself. If I'm too fast, let me know. So I'm going to, in Google, go and type Swiss model expressing, or I have given in the manual, I have given the link clearly. So if you click on to the link, it will directly go to this particular page. So here you can see start modeling option. So this is the home page of Swiss model, where you can take the sequence and paste it over here. So I'm going to copy the sequence from here and I'm going to paste the sequence. And once I paste the sequence, I can give my email ID if required. I'm going to type the project name alone, GLUT4. So email ID, it is not mandatory, so I'm not giving here any email ID. I'm going to click on to the option search for the template. So depend upon your server or a sequence length, it sometimes it take time, or once it run, it will give you the template. So any doubt till now? So till it run, if you have any queries, let me answer.
Ma'am, kindly repeat it again, ma'am.
Yes. Yes, ma'am.
Can you repeat again, ma'am?
Swiss model, ma'am.
Yes, ma'am.
Okay, fine. Okay. So, uh, so before going into Swiss model, we require. So should I repeat from sequence retrieval or directly shall I go to Swiss model alone?
Swiss model alone, ma'am.
Okay, fine. So in Google, go and type Swiss model. So click on to the option Swiss model and click on to the option start modeling. Okay, somebody asked for from the sequence level, so I'm going from the sequence level itself. So as usual, we know UniProt. So I'm going to search any one particular protein without the structure. So I know this particular protein doesn't have the structure, so I'm using the GLUT4 option. So I'm going to click on to the option search, and so you have to click on to the entry information. So if you want the structure, sequence, so here you have different type of option. You can click here the sequence directly, and once you click the sequence over here, here you have a FASTA option where you can click and get the FASTA format of your sequence. So I'm going to copy this FASTA format sequence and I'm going to give this as an input to the Swiss model. Okay, and so in Swiss model, I'm going to, so I have told you how to open the Swiss model. Go to Google and type Swiss model. So the first link would be this particular page. So once you open this particular page, click on to the option start modeling and you have to paste the sequence over here. Once you paste the sequence, so you have to give the sequence title or project title GLUT4 and click on to the option search template. So this is what I have already told. Here I have already searched for template. It has searched and it gave some result over here. So this GMQE and GSQE is the score. I have told you this score should be nearer to 1. So when you going to publish this model. So you're going to say that you have predicted this particular structure using Swiss model, and you need to mention this GMQE score. I have given a detailed explanation about this GMQE score in the manual itself. So you can see the definition, all those things over there. So I'm not going depth into definition. And here you can see the query coverage, where you can see initially little white region and last you have white region. That is the region which was not modeled. The starting and ending of the protein was not here. So for this only we will check the domain detail, whether that particular region was biologically important or not. So once I choose, so here this one has 58.99 and second one has 65.92. But the query coverage here varies, so only it was ranked the second. So I'm going to click both the models. So I'm going to click first option and second option. So here I'm going to build two models. So I'm going to click on to the option build model. So here you can see here this modeling was started. So here you can see the modeled structure over here. The first one is your model one. It was built using this particular template. This template is nothing but it was already available protein structure which was present in the database. It has a query coverage of over 58.99%. So here you have this arrow. No? If you click on to this arrow, you can see the alignment for your modeled structure and the template. So you can see here some score. No, this score was calculated based upon this particular alignment. If the more the similar amino acid, the score would be more. If there was mismatch or more the identical residues, if it is more, the percentage would be more, and similar residues, that is similar category of amino acid. If there was more mismatch, the score would be less. So this is how the structure would be predicted. Here we will check for this particular score and this particular value. And additional to this, so here we have two different structure. One is this and one is this. This particular structure has more Q mean value. That is why it was listed first. So we can check both the option. So once you find you can check for structural assessment to check the Ramachandran plot. So I'm going to click on to this structural assessment link, where it shows the Ramachandran plot for this particular structure. You can see here, so it is a membrane protein. It was showing this membrane region was also here. Here you can see here, these are all the dots or the amino acid present in this particular protein. So there was no regions present in no amino acids present in the disallowed region, and here you can see 98.5% was present in the favored region. You can put this in your, but even though some amino acid, it doesn't have good bond angles, that was also shown here. So this should not be more, and bonds and angles was present with these kind of amino acid, that was also listed over here. And you have to mainly check there should not be any residues that present in the outliers. So here it shows 0.65% was present in the outliers, but even though here it was no region disallowed, we can go for this data and you can proceed with this structure. And mostly 98.5% was present in the favored region, so we can take this structure for our analysis purpose. So once you verified with the Ramachandran plot, once we have done with the Ramachandran plot, we can download that particular structure and we can keep it for our docking analysis. So clear? Any doubts? Any doubts? Okay.
Hi, good afternoon, Miss Shubha.
Yes, ma'am.
Like,
Yes.
if it is going to be an unknown protein, then how we proceed about it?
If it is mostly unknown protein, we do blast usually. So if you have a protein sequence and you don't know the protein name, in that case, we do the blast and we try to find similar protein name or that if we do means we have the alignment for the protein and based upon the alignment if it was more similarity, the percentage was 99-100% means it was the same protein whatever already present in the database. If the identity was very less, it means it was a new protein, it doesn't have any data. So you can't like for every protein there would be similar fold would be present. Even though the exact similarity percentage was not there, the similar fold would be there. So based upon the fold, we can go for, so if we can't find the sequence identity, so if the sequence identity was less than 40%, then in that case we can't go for this particular model because there would be more amino acid, it would be fall in the outlier region. Then in that case we can't choose this particular methodology, this particular modeling technique. So if the similarity was good only, we can go for homology modeling. If the similarity was less than 40%, we need to go for next method, fold recognition method. If the similarity was between 20 to 40, you can use that fold recognition method also. If the similarity was less than 20% only, we need to go for ab initio method. That last method which I told in the protein structure prediction. Clear? Am I clear?
Yeah, thank you. Yeah.
Okay, and so here once I check the Ramachandran plot, I have to click on to this model one, this icon, and click on to PDB format. If I click this PDB format, this file would be automatically downloaded. So once I completed once I downloaded the file, I can click the file and you visualize using Rasmal. So here you can see the structure file. Um, Based upon the color or based upon structure cartoon model, you can see here this is completely helical structure, I mean alpha helix, and only here it was a beta sheet. This blue color is your coils and white color is your loop region. So am I clear? So in Rasmol, if you want to change anything like background or increase, so you can't zoom it using mouse. So in Rasmol, if you want to change your background color, you need to type background and white in this command box would be open along with the Rasmol. So once you type background white, the color will change, and percentage if you want to increase the zooming in percentage. I mean you want to zoom in, you want to zoom the structure. So you have to click zoom and give 200 means it will double actually. So you can see here. So these are all the small option in Rasmol, for just for the publication purpose, we require clarity of clear image. So in that case, if you want to zoom in, so you can use zoom option. If you want to change the background color, you can use background, whatever color you require, you can do. So this is about protein structure prediction. So am I clear? Any doubts in this? Shall I move to ligand preparation? Shall I move to ligand preparation?
Yes, ma'am.
Yes. Okay. So let me go with ligand preparation now. Let me start the ligand preparation methodology. Before that,
The voice is not audible, ma'am.
yeah, yes, ma'am. Yes, I'll open the PPT in just a minute. So let me start with ligand preparation. So before going into ligand preparation, let me give a brief introduction about how to choose compound and how to take compounds. Um, Presentation is visible, no? Okay. So ligands are not. So first we saw about protein preparation. So now we are going to move about ligand preparation for docking. So ligands are nothing but it was called as your guest molecule or a key molecule where it will go and bind to the receptor, that is your protein. So ligands are the substance, that is a chemical molecule or a small molecule or a biomolecule used to serve for the biological purpose. So usually the ligands usually produce, so once it go and bound to a protein, it will fit like a lock and key. So that is the usual concept of docking, where it will go and bound to an active site of your protein and it will fit into a protein, and it will change some signal in your protein, or it will bound to a protein and it will inactivate or downregulate or upregulate your mechanism of your protein. That is the actual purpose of your any compound. So why we are not telling here drug is once it was approved by the process FDA, then only we call any molecule as drug. So before that we call in bioinformatical term, we call them as ligand or a chemical molecule or a small molecule or an inhibitor or an activator, like that we call them. And what were all the lead finding or a lead generation approach? So usually, so anything we use, we have many techniques to identify the lead compound, usually older techniques or animal model as a human disease, or random screening approach or ethnobiological approach. So ethnobiological approach means plant-based bioactive compounds and modification of the known drugs to develop a new drug. So you are modifying some side chain and you are producing some drugs, and so these are all the usual approach to screen for a lead compound. So these are all the some of the example of already existing drug. So this is atropine belladonna. This in this particular plant they have isolated the atropine drug, and Papaver somniferum. They have isolated morphine from this particular plant source or this particular flower, and Raufolia serpentia. This flower usually we will see mostly in the campuses or in gardens. We can see this particular flower where they isolated Vesperin from this particular flower and digioxin from Digitalis lantana. So these are all some of the examples from the plant-based lead compounds. And animal, so even from animal also you can isolate the compound. From marine based compounds you can isolate from the shells you can isolate some compound, and from snake also you can isolate some of the toxins. That was also because you know very well all the drugs were toxic, but it should be used in a limited levels. And next, from the microorganism also we can isolate some of the lead compounds. So these are all the already isolated drugs from the microorganism. So from penicillin was isolated from Streptomyces mutatum, and chloramphenicol from Streptomyces venezuelae, and Griseofulvin from Penicillin griseofulvum, streptomycin from Streptomyces griseus, and neomycin from Streptomyces grandii. So these were all the different drugs example or a lead molecule example. So usually ligand or a compound database, usually we have PubChem database to retrieve the compound, and drug bank database where you have, so PubChem is the most common database. We can go and search for the compound structure and biological activity and the assay. And so usually in PubChem, these were the recent data I have got from PubChem. It was around 111 million compounds present in it, and 277 million substances, and 293 million bioactivities were reported, and 33 million literatures were available, and 28 million patents were available. So drug bank, it is another database where you can find the complete information about drug and drug targets. This was maintained by University of Alberta. It was present in the Canada. So these were the two main drug compound databases we use. Today we are going to see about PubChem. Okay, so PubChem, usually we will go for this link to download the compound from the PubChem or a compound details from this particular database. This is the home page of PubChem where you will go and search for the compound name. So it could be any of the flavonoid or any phenolic compound, polyphenolic compound, whatever it is, or you have something from your GCMS result that you need to, so in that case you can't find some structure won't be available in database. In that case you have to draw the structure, then you can go for the structure analysis. And for chemist, some structure won't be available because they would have modified some side chain, they would have got through NMR, so they would have got in a drawing format something. So in that case, you can draw the structure using any one of the chemical drawing tools and you can convert it into the 3D format. So usually we type here and click on to the option search, and so once you search, you will get the entry like this. So once you click over here, it will go to the entry page where it will have all the information about the compound, which I will definitely show practically. So usually we take smiles format from here to draw the structure in the required format, whatever we require. We can try manually also. I'll just show you how to draw the structure also. For short form, we require the SMILES format, and for any drug likeliness prediction or a molecular property prediction, we require this particular format. That is why I highlighted this. I highlighted this particular format. So you can copy this format for any structure, whatever you are taking. You can keep this for your further purpose, for the analysis purpose. So for protein, we are taking sequences. Similarly, for compound, we require SMILES format for analysis. And so usually this is ACD Chem Sketch where you can draw the structure, or using the shortcut you can, using the smiles format also you can import the structure. So in that case you have to go to tools, generate structure from smiles, and you have to paste the smiles format over here, and it will show some box like this. Once you click OK, your structure would become here like this. You can see structure. This is your 2D format of the structure. So we got the 2D format over here, but we require 3D format for the analysis purpose or a docking purpose. So for docking Argus Lab, whatever we are going to use, we require MDL mole format for Argus Lab docking. So for that we require 3D format. So in ACD labs, if you click over the ACD labs over here, you have 3D viewer. So if you click on to the 3D viewer option, it will open in this format. So the background would be usually black. So using the color option, you can change the color of the background. And here you have different depiction line models. Here you can see my arrow, I think so. In line model, scaled ball and stick model, or different models would be available. So this is a general process. Once you have a 3D format over here, you can also optimize your structure by clicking this arrow mark. So you can, I hope everyone see this arrow mark where you have this symbol. So if you, this is nothing but 3D optimization. So usually when we draw the structure, it would be in the rigid form. So when you click on to this optimization, that fold would be happening. Little bit your structure would get folded. So if the structure was very much rigid, so it would be like, so you know very well there would be some rotatable bond where you will be the protein, the compound would be flexible enough to bind to the active site cavity of your protein. So that is how your protein compound will go and bind with your protein. And so here, once you done with your 3D model, you have to go to save option, file save as, and here you have to choose MDL mole format, and you have to click on to the option save, and you have to save the structure. So there was one more option available. This software you require MDL mole format, so we actually done with the ChemSketch. So you can using that MDL mole format, you can convert this format, MDL mole format, to PDB format using any one of the converter tool. Actually, this was not required for us, but generally you also have to use the conversion tools also for specific format we require. So here you can choose MDL mode format, and if you click on to the option convert to PDB format, you will get the output over here. You can copy this structural details and save it in WordPad in .pdb format. So this is how we usually save the compound structure. So shall we move to the practical session? Am I clear? Any doubts in the presentation? Any doubts in the presentation? Shall I move to the practical session? Okay, fine. One second, let me start with PubChem database. So any one drug example, can you type in your chat box? Any one small molecule or a compound name? Or if you want to work with any compound, can you tell me any one compound name? Yeah, definitely I'll share my PPT. So can you share any compound name in your chat box? Okay, sulfur saline. So in Google, go and type PubChem, or in manual I have given the link for PubChem. You can go with the link also. Um, so, so I'll just type sulfur saline. Celestine. Okay. So this is the structure of the compound, whatever sir has posted. So I'm clicking onto this option. So once I'll search, I'll get the different option. So here you can see list of option. So mostly our query would be first option. In some cases, if you have multiple options, you can check, you can cross check with your molecular weight and molecular formula. Hope everyone know the molecular weight and molecular formula before going into whatever compound you require. I hope everyone will know your molecular weight. It would be given in your GCMS or LCMS result, or when you synthesis, you know your molecular weight. So you cross check with your molecular weight and molecular formula. And the CID is important for publication purpose. Suppose if you're going to publish this compound, so you can mention the CID, PubChem ID, which was also required for publication. And this is your PubChem ID. Then comes your structure, and definitely the chemical safety, which was already reported, was given in this particular chemical safety icon. So this particular compound, it was a hinted, it was used in a more amount, it was also caused health hazard. And you can see here the molecular formula and different names. So if you click more names, so you can see here the IUPAC name, different names of the compound. So mostly in GCMS results, we get these kinds of names. So when you search over here, you can find, but you will have the short names also, or a depositor names also. So whatever it was easy to use, you can use that. And you can find molecular weight over here, later which date it was initially created and which date it was modified recently, and what were all the different mechanism of action of this particular compound. And this is a 2D structure of it, and this is a 3D structure where you can rotate and check the structure of this compound. And uh, first let me try to draw this particular structure. I think I have downloaded ACED Labs. If anybody, I hope everyone have installed your softwares. Anyone have any issue with any software? Anyone have any issue with your softwares, installation or download? Okay, I hope not. So let me install ChemSketch. So I'll just first draw and show you, then I'll show you how to use the shortcut, because if the protein, the compound doesn't have the structure you need to draw, then you can convert it into the required format. Okay, so ChemSketch was installed. So once the ChemSketch was installed, it won't be available in desktop. So here you can type in the search box, you can type ChemSketch, or using in programs you will be having this icon. So hope everyone see this icon. So click on to that, it will open. So it will show like this. So don't go to any other link, just click OK. Click to the option cancel, then click to the option OK. Okay. So here you can draw the chemical structure of the molecule. Let me have the screen side by side and I'll try to draw the structure. Hope the screen was visible. So here the structure, I think here it has 3 benzene ring. So usually the compound have the library over here. You can see, I hope everyone see the benzene ring over here. Can you able to see the benzene ring? So once you selected the benzene ring, click on to the option C in the left side. Okay. So here the next part is here. I'm going to put N over here. So I'm going to put here and here. So I'm going to choose the option N. So I'm going to join this tool. So this is how I should draw. If I need double bond, I have to click here again. And next I'm going to include sulfur group over here, sulfur group and this SONH, SO2NH group. So it was actually linked in this way. So for S I need to select S from here and double bond O. So O atom I have to select. And I'm going to link this with NH. And here you have N group. So today everyone practice this. If you have any queries, you can ask me tomorrow also. And you have OH group. And you have one more C group here. And OH group and double bond O. And I have to check the bonds also. So here 3 bond, it was okay. Here it was okay. And here it was not okay because it was in this way. So if you do any mistake, you can use Control Z or you can actually use this eraser option. Yeah, definitely we'll do it again. You can use this eraser option. I'll repeat once again. First I'll complete and just show you the formula and all these things. So I have changed the bond. So now I will just cross check with the molecular formula and molecular weight. The 398 is the molecular weight, and this molecular formula is C18H14N4O5S. So here I think everyone can see here at the end over here at the last line. So you can see C18H14N4O5S, and here you can see the molecular formula 398. So because we draw it randomly, So you have to clean the structure. So using the tools clean structure option, you can clean the structure. Now actually it would be very neat and clean, because we are drawing it, so we can put bond here and there, over there, here and there. So that is why we need to use the option tools clean structure to get the neat structure. Okay, so I'll repeat once again how to do this. So first, so I'll delete this. Uh, so you have 3 benzene rings, so I am placing 3 benzene ring over here, and next there would be in between you have one nitrogen group linked in this way. Okay, and next part is your S group, sulfur group, and nitrogen group over here was linked. So I am clicking here. In the left side you have different atom name. In the right side you have benzene rings and other side chains were available. So based on this you can choose, and from here you have to join here. And for sulfur you need to have a 2 double bond O. So you have to O atom over here. So I have drawn correctly, I think so. Here one NH group were pressed. Well, here one NH group were here, and here one O group, and C, OH. So usually the OH will come once you click. If you click bond over here, it will be changed as double bond. I hope I have done with the structure, and you have to check the bond alone. So if you click over that line, it will show like box. If at that time if you click it, the bond will appear. So now I have drawn the structure. So hope everyone. So once you drawn the structure, how to delete the bond? So if you click on to that box again, the bond will go. And if you want to delete one particular part of the structure, you need to use this delete option here in the below the document icon. You have delete option. So if I don't want this particular oxygen atom, if I delete, it will go. So once you're done with this, go to tools, click on to the option clean structure. Clear? Somebody has message, cannot see the shared screen. Can you pin my name? Actually, for everyone's screen was visible, no? Okay.
Yes, it's visible, ma'am.
Yes, okay, thank you. Okay, so once you draw the structure, so this is how we will draw any chemical structure, and if you want any other atom apart from that, you have options over here to choose all those things, so that you can try later also. So let me tell one more option how to use using the smiles option, how to draw the structure. So you can take the canonical smiles from here, you can copy this entire line without space. So you should not have additional spacing characters, so you have to copy the smiles part alone. You click onto the option copy and go to the tools, generate structure from smiles, and paste the option over here, paste the smiles format over here and click here. It will click and paste it over here. The actual structure will come over here. Am I clear? So this is easy option where you can use the smiles and bring your structure over here. Tools, generate structure from smiles, paste the smiles over here and click on to the option OK and paste it over here, you will get the structure. So once you're done with this, you can go for 3D option, ACD Labs, 3D Viewer, S. So if it doesn't appear, the empty screen appear, here you have ChemSketch, copy to ChemSketch 3D and 3D optimization. So click on to the option ChemSketch
Ma'am,
and here,
excuse me.
yes,
Ma'am, can you
yes,
repeat that canonical structure, ma'am?
canonical structure, yeah. Okay. So I'll delete this. So you have to click on to the option Tools, Generate, Structure from Smiles and paste the smiles over here and click on to the option OK. And it will appear like light color. So once you click the arrow, it will actually get pasted. Clear?
Okay, ma'am. Thank you, ma'am.
Okay. So once you done, already we have clicked ACD Lab 3D Viewer option. It was available here. You can click here, copy to 3D. In the key, here you have 1, 2, 3. No. So in second option, you have copy to 3D, where it was actually copied to the three-dimensional format. So here you have line notation, uh, stick model, ball and stick model, space fill model. So these were different models, cartoon model. So usually for publication purpose, usually this would be clear based upon the atom color, the different atoms would appear. Carbon atoms were appearing in blue, light blue color, nitrogen atom dark blue color, oxygen atom red color and sulfur atom yellow color. So once you done with this, you have to click 3D optimization where the hydrogen bonds were added to the structure and the structure would get optimized. You can see the folding form of the structure. I think everyone able to see this. The structure was getting little bit folded. Once you're done with this, click on to the option File, Save As. You can choose MDL mole format. You can create a folder for protein and your compound. Um, you can create a folder in desktop. I'll create a folder called docking. I'll just put the name of the compound. I think it was sulfasen. So I'll put the name of the compound and save it as MDL mole format. That is the extension it should have .mol and click on to the option save. So now your compound was saved in MDL mole format. So now you can see here. So here the compound was saved in MDL mole format. And initially we have the protein structure also with us. We have downloaded a protein, 2 different protein and we have model 1 protein. So with this we perform docking by tomorrow. So till now am I clear? Any doubts in this? Yes.
What's the difference between smiles and canonical smiles?
The way of notation, canonical smiles usually, canonical smiles is a smiles format. That was not any difference. Smiles, like canonical is the name given over here. So it was actual smiles format only. So smiles is a simplified molecular input linear entry system where you can have, based upon this bonding, the structure would be actually the software would take the, so using this format, the software will convert this into a structure format. Clear?
Okay, ma'am. Thank you.
Okay. Any other doubt? So this is how we take the compound data. Yes? Yes, any doubts? So apart from that, you can get lots of compound related information over here, like compound computed. So in PubChem itself, Yeah, you have many other softwares. You have for drawing, you have ChemDraw software. Um, okay, someone unable to download the 3D one. So let me repeat once again this ChemSketch alone using the smiles format. Let me start from the first, then only I can tell you clearly. So I'll open the ChemSketch icon. Okay, okay, cancel, okay. And using the canonical smiles, I try to generate the 2D structure. Okay, so here you have ACD Labs 3D viewer option, right? So click on to that. So initially it won't show anything. So it will be a blank screen. So I told you to go to ChemSketch in the downstairs. In the left side you have ChemSketch icon. ChemSketch, you have to click on there and click on to the option copy to 3D. Okay, so once you click on to the option copy to 3D, your compound structure would be available. Then you have to click on to the option 3D optimization. If you don't have that option, then you might haven't clicked to this ACD Labs 3D viewer. So you check whether the stick was available in your the stick mark was available in your in your window. So once you loaded over here, you have to optimize and save it. Okay. So, am I clear about structure retrieval and compound structure prediction? Any doubts in this? And I've told regarding the schems, sorry, you can find some of the property related information from this PubChem. Usually all the chemical, physiochemical parameters related to the compound, you can get it from here. Tomorrow let me teach about ADME, how to calculate ADME parameters and ADME parameters and Lipinski rule, that is your physio, like a log P value, hydrogen bond donor, acceptors, rotatable bond. So each has specific role for its property in docking. So all these things we can see tomorrow. And docking methodology using Argus lab, that will also we can see by tomorrow. So if you have any other doubts, let me clarify. Otherwise, we can wind up this session. You can put it in your chat box. Let me clarify all your doubts. Vijayalakshmi have asked about could you give me a pathway how to proceed if we have unknown compound extract, how to proceed with these tools. So if you have unknown compound extract, that compound would have particular activity, right? It could be antimicrobial activity or anti-cancer activity. Even though it was your unknown compound extract, that extract would have certain activity, I hope so. So that based upon that activity, you can choose a target. Suppose if your extract have anti-diabetic activity, for example, or if it is completely unknown extract, means you have to perform some assay to know the activity of the compound. No other way for a completely unknown compound extract. Like if you doesn't know the any activity for the particular plant source, then you have to do it randomly, antimicrobial or antifungal or anti-diabetic or anti-cancer activity. Then you have to based upon the activity, you can go and find from the literature what protein have to be chosen, and you have to study the pathway pertaining to that particular disease, and based on that you can find the target, and later you can find the compound structure using this one.
Yeah, okay, ma'am.
Okay.
It's an antimicrobial activity.
If it is antimicrobial activity, then which microbe it was antibacterial, which bacteria it was much effective, then you have to refine which bacteria. For example, if it is staphylococcus aureus, you have to choose a compound, a target pertaining to staphylococcus aureus.
Okay, okay, ma'am.
Okay, so in some cases, how to prepare multiple ligand means you have to draw each ligand and you have to save it separately.
Okay, so we can docket each ligand and know the.
Each ligand we can dock separately, or you have one thing called virtual screening. At the same time you can give multiple ligand input and do, but I suggest it, if we do it individually we can get better result, because some protein might have more binding energy, but it may have good interaction. So usually when you do multiple ligands simultaneously, based upon the binding energy you choose one particular confirmation and you proceed. Usually you proceed with interaction analysis, but when we do individually we check each interaction separately. Based on that you can, based upon the interaction also we can give some conclusion, based upon the binding energy also we can give some conclusion. I drawn the chemical structure in drug bank, so that structure used to do the docking, madam. I don't know. I drawn the chemical structure in drug bank. Yeah, you can draw that also. That won't be a problem. Your if your ligand was your short peptide, then you can go for peptide protein docking. That was also possible, but I don't think this software would be applicable for that. This is for your ligand and protein docking. There was some software called Galaxy PepDoc was available for this these kind of usually when we go for antigen antibody, suppose for if you take for COVID, in that case we find some antigenic epitopic region that should bound to receptors in the human target. So for that, in that case we can go for uh, peptide protein docking. So for that there would be a separate service for a protein peptide docking. Definitely your short peptides also could be used as a ligand. PFAM, good. I have actually opened PFAM, but I couldn't do it that time. Let me try PFAM once again. With PFAM we wind up. If it is not working, we'll definitely tell you tomorrow. It was actually working. So let me go for sequence search. So in PFAM, so this is the home page of PFAM where you can go for domain analysis to identify which part of your protein is important. You can go for this particular analysis. So let me take any one protein pasta sequence and click on to the option sequence search and click on to the option go. There are currently too many jobs in the sequence search queue, so please try little while. Okay. So there was one more option where you can use this UniProt ID also. So here you have accession number option no. So you put that and click on to the option go. So because I think many of you were trying, or I don't know that, but so it was not showing. So that is why I used this option. So here you can see domain over here. This domain name is sugar TR. So it was related to diabetic protein. It was something like this. So if you click on to this detail, you can study about this particular sugar TR domain. This sugar TR domain, sugar transporters, that is glucose transporter, no? So they have mentioned it as sugar transporter. So here you have disorder region was present in 2 to 13 position. So the important position here is 27 to 483. The actual length of GLUT4 is 509. So initially when we modeled the GLUT4 structure, we saw the initial and final part was missing. So that was not much important part. So that is why the actual exact region is 27 to 483 was your domain region. So that part alone was got modeled using the modeling. So when you write about domain in the using PFAM, we found this particular domain from this position to this position. That was also, that part of the structure was available when we model. So like that we have to write when we write an article. So am I clear? Any other doubts, any other queries? Any doubts? Okay. So if you want to, like if you, for tomorrow's session, you try to take one particular protein and you similarly you try to find any compound. So if you don't know compound name, like I have given some example in the manual, you can prepare that compound for that purpose. And if you have doubts while preparing, you can put it in your WhatsApp group also. Let me clear that during evening also. So tomorrow we proceed with the practical session for drug likeliness prediction and molecular docking introduction. And tomorrow there was one more additional session by Benita Benoy. She'll perform that CSAR, that is virtual screening using the commercial software. That software link also we'll share by today. That session would be morning first session. That would be followed by my session where we'll perform all the property related information, and finally we'll proceed with docking and visualization. So shall we wind up the session? Yeah, definitely we'll share the video, no issues. And not by today, let me share by end of the session. Okay. Okay. So if you have any queries, let me answer. Otherwise, I'll wind up the session.
Ma'am, I couldn't download PyMol software, ma'am. Is there any other
Pymol
link?
software. Pymol, you check your system property whether it was 64 or 32 bit.
The
They
64 only, ma'am.
64 only. You couldn't install it.
Yes, the
What
page
you
is not
just
coming. Website
the
itself didn't open,
website
ma'am.
itself it is not coming. Let me check the website. Just a minute. Okay. I cannot. So Argus Lab, it would be open as zipped file. So you have to extract the files and try to install. So Argus Lab, I have also downloaded. So it will be usually open. It will be. So if you don't have extractor, try to use any extractor tool to extract the files. Okay. Once you extract the files, so it would be like this. So here you have setup file. Once you click on to the setup file, it will get installed. Yeah, tomorrow I'll be a little bit slower, Risha. Let me see. So any other queries? Venkatraman, you try to install Argus Lab in this way. Swiss model, I mean PyMol, in Google you go and type PyMol ma.
Okay, ma'am.
Um, So this spymol.org, you click on to that particular link. Here you have download now, you can click on to that particular option. You can choose Windows exe installer, or if
Okay,
you
ma'am.
want to, or you can download zipped archive and try to extract and use it. Okay. If you have
Okay, ma'am.
any error
Thank
while
you, ma'am.
installing, let me know. We'll definitely clarify. Okay. So let me wind
Okay,
up the
ma'am. Thank you,
session.
ma'am.
Okay. Thank you.
Thank you, ma'am, for your valuable session. I hope everyone had a wonderful session today. I extend my gratitude to thank today's speaker, Dr. Girinath sir, for his valuable information, and I thank every faculty and the participants. I would like to end this day with a quote: "Knowledge has beginning but no end." As the saying goes, we will continue our workshop tomorrow morning by 9:30 a.m. Thank you all. Thank you, ma'am.
Thank you, thank you, participants. Tomorrow join by 9:20 a.m. Thank you all. Any queries, you can post it in your WhatsApp group. So those who didn't fill the certificate form, kindly fill that form. I'll just put the names in the WhatsApp group. Those who didn't fill the form, kindly those candidates fill the form.